Hi, everybody. Today we are going to be talking about the kingdoms of life. So this is it. This is the big one for this unit. This is the major focus. The other lessons after this are probably not going to be as focused as the kingdoms is really the broad category we really want to dig into in this unit. So, life, the broadest category, incorporates all living things. We've discussed this already before. It's based on the cells, the genetics, morphology, other characteristics. Then we take that and we divide that into 7 other kingdoms. If we looked just at the cells alone, we could even divide it into 3 parts, 3 kingdoms. Previously, these were called the 3 domains of life, and those are the archaea, the bacteria, and the eukaryotic. So there are other names that we've seen mentioned. I think we brought them up in class even at one point. But there's sometimes this is known as eubacteria. I think if you look at the tree of life, uh, and then this is sometimes called archaea bacteria. That's another possible kingdom. So those are other names for it. Eukaryotic is also known as eukarya, and eukaryotes, eukarya. Sometimes these organisms are known as the eukaryotes. So these are all sort of synonyms. So I just want to put these out there into the world because these are sort of the synonyms that we might find if we're looking for other resources. I will try to continue to use bacteria and archaea to explain when I'm talking about these, and I'll try to use the eukaryotic as much as I can. But if you do go out there looking for videos, looking for resources outside of this, you might find them referenced as archaeobacteria, eubacteria, and eukarya. All right. So previously, these were the 3 domains, and all of the organisms in these kingdoms have these shared traits. So archaeobacteria have an ether-linked lipid. Don't have to know what that is, other than to remember that's called the ether-linked lipid. It has something to do with oxygen. That's all you need to know. And we call this ether-linked lipid pseudopeptidoglycan. So that's a word you just kind of have to learn, and you know, and you need to learn that they're ether-linked lipids. So if I am going to be referencing something, I might be using these words. Bacteria are ester-linked, so slightly different lipids, and they are made with peptidoglycan, not pseudopeptidoglycan. And eukaryotic organisms have ester-linked lipids, but they also have various structures. They don't just use the peptidoglycan. So let's change the color over here to show that sort of difference. So we see the archaeobacteria are ether-linked, but the other 2 are ester-linked. And we do have a pseudopeptidoglycan that's unique to the archaeobacteria. Originally, archaeobacteria or archaea and bacteria were together. We didn't know that they were different at the beginning of when we started studying life. Um, but then we found this difference in the cell membrane that was really important. Later, they started studying the chromosomes, and chromosomes being like all the building blocks of the cell follow the chromosome. So the type of chromosome was pretty important. And circular chromosomes are found in archaea, circular chromosomes are found in eukarya or in bacteria, but in eukarya we find linear chromosomes. So that means straight lines. So these chromosomes, they kind of usually there's sort of these long chains, but they are really long. Like we are talking like super long chains here, and they typically kind of just stretch out. You could stretch them from one end to the other. In in the circular chromosomes, we have a completed circle here, and the DNA kind of just curls around. And they're much shorter. Like they don't have as much genetic information as eukarya has, because eukarya has has all of our multicellular organisms. So the key here, though, was that the key observation that evolutionary biologists enjoyed was a use of eukarya-like transcription and translation. So that made this interesting connection. Although the cells appeared on the outside to have ether-linked lipids and pseudopeptidoglycan, which suggested they were more like bacteria, the transcription, the actual way that information was handled and processed in the cell, was much more similar to the eukaryotes. And so that's where they said, hey, I think archaea and bacteria are related to each other, but bacteria are not an ancestor to eukarya. Archaea are an ancestor to eukarya, or archaea shares an ancestor with eukarya, because the processing of the DNA is such an essential factor in sort of uh, determining the type of cell. Now, eukarya have membrane-bound organelles and nucleuses, so that's a really key feature. We don't see that in the bacteria or the archaea. They have neither of those things. We get various metabolisms, including things that are like methanogenesis, which is a type of consuming methane. It's not the only type, though, uh, but this is unique to the eukarya. So this is only one, as you know, this is only one example. They have all the other ones. They have aerobic, they have anaerobic, they have fermentation, they have all that other stuff as well as this. So they also all of these. So this is the only unique one. I'm going to get rid of this, but I'm going to say that's the only unique. They get this and methanogenesis. And then we also have on the other side, we have the photosynthesis, which we do see in some bacteria. There are some photosynthetic bacteria, so we do have, something that's shared here. Metabolism is all over the place. We can't say that it's not a really good defining feature between the three groups. And then for reproduction, we have three different possibilities, or we have the three groups, and we have asexual reproduction in all three. But we only find sexual reproduction, true sexual reproduction, in eukarya, because that's really where we see defined sexes versus the single-celled organisms, which do not have that case. They also have that horizontal gene transfer I discussed earlier, where we are going to have two of these bacteria things, or archaeans, and they are going to connect their cilia. And when the cilia connect, they can actually transfer information through their cilia genes. And that's really weird, but it is something that they can do, only they can do. Okay, so we're going to break down each of these kingdoms really briefly, and that's it. This is the important part from this unit. We will do another video, a follow-up video for the rest of the unit, but those are extra. We're not really going to focus in on any of them. We're probably going to do a project with the other parts of this booklet. So we have kingdom bacteria. All members of domain bacteria are in kingdom bacteria. We didn't talk about domains, but I did mention that these are the three domains that we have. So property, these are also, I don't know, do I want to make a mention of it? These are also the domains. So in an older style, we've seen some images that have that. All right, so they're all prokaryotic. No nucleus. They all have cell walls that are made of peptidoglycan, which is some sort of a mesh of sugar and amino acids. They have ester-linked lipids. They have circular chromosomes. They have all sorts of types of fermentation and metabolism. They have asexual reproduction. All right. So we also have kingdom archaea, and that is all the same factors. They previously called domain archaea, and they are more ancient than bacteria in some cases. This is something that's been debated now. So the modern bacteria that we see today uh, are much different than the original bacteria, whereas archaea is actually a lot similar. Even the organisms that are living today are not like you're not the organisms living today are not billions of years old, but they share more characteristics with their ancient ancestry. And that's partially because of the way that these organisms exist. So they have these ancient ties. They're closely related to eukaryotic ancestry. They have prokaryotic cells, but the walls are not made of peptidoglycan. They have ether-linked lipids as opposed to ester-linked lipids. They have the circular chromosomes. They undergo genetic processes of transcription translation, but those processes share features with eukaryotic cells. That's one of those key features that I really want to attract your attention to. So this is that eukaryotic similarity. Uh, and they undergo asexual reproduction, and they are some of the oldest organisms living on Earth. They've had the least change over time. Now, one thing that is missing here, which I must have overlooked, is that these are also known as extremophiles, or at least many of the, let's go like many archaea species are known as extremophiles. They live in some of the most extreme environments in the world. Extreme. And so they live in salt pools, volcanic vents, uh, this digestive systems where there is a lack of oxygen, and more. So this is a very unique system, and this unique ecology leads this unique ecological niches lead to their lack of evolution over time. Something we might discuss again when we get back to evolution. But if you're something that is isolated, they live in these super extreme environments. There's not a lot of competition. They only compete with themselves. So the amount of genetic change they undergo is very small over that time. That's what I want to get at there. And lots of people talk about, lots of times you'll see in videos about archaea, they'll talk a lot about the extremophile nature and how they're sort of these interesting ancient organisms that are left lying around. All right, so the next kingdom we're going to talk about is the protozoa. And again, I'm going to mention that we might have some trouble finding information about just protozoa. You might find it called protist or protista. And that's because that's the older naming convention, especially popular. The 60s naming convention was extremely popular, and a lot of textbooks still have it. They don't have organs. So these are still our single-celled or colonial or multicellular. So unicellular, colonial, or multicellular. So there's a couple of different namings here. Maybe we should define those real quick. So unicellular, one cell makes up the organism or the individual. Colonial organisms have single cell individuals, but they have low survival rates if not in a group of individuals that work together to work together. So that's a colony. If you wanted to go on a macro scale, there is an argument made about ants. So ants are thought of as colonial organisms as well, except they have multicellular bodies, but an individual ant living by itself can't actually survive. They can't find food properly. They can't reproduce by themselves. They need the rest of the colony. So that's called like sometimes known as a superorganism. Um, Colonial organisms and the protists, they are these sort of groupings of single cells, each technically an individual, but if you took one cell and threw it out in the ocean by itself, it likely would not be able to gather food efficiently and not be able to take the food down. By working in a colony, those cells can function together and do some of those tasks together. Multicellular, of course, is what you might be most cellular is what you might be most familiar with. This means that the individual is made of two or more cells, and not much else to say about that. I was just wanted to raise the idea of the colonial organism because we haven't talked about those before, probably. They often live in moist environments. We don't the influence of water on protists is really, really important. And they get organized into two categories. The animal-like protists, which are also known as protozoans. They're heterotrophs, meaning they eat others. They don't produce their own energy, much like bacteria as well. Many bacteria are heterotrophs. And we see examples like the amoeba or the paramecium, which are these sort of moving organs, um, or moving cells that can, interact with each other. You can have amoeba that hunt paramecium for food. You can have there's a type of animal-like protist that actually engulfs a chromista inside of it to provide itself energy and other things like that. We have also fungus-like protists, which include the slime molds and mildews, and I'm sure I'm gonna link to a very engaging video about slime molds and how interesting those are. They're sort of a single cell filled with nucleuses. So I'm just gonna put in heterotroph here as part of our language. So a heterotroph, it's short for eats other. So this is an organism that that gathers its energy by consuming other organisms. We contrast that with autotroph, uh, and it's a self-eater. That's sort of the rough self-eat. I don't know, something like that. I don't know the exact. That's sort of the origin of that word, but it's an organism that can produce its own energy, like plants. So I'm gonna have to clean this up a little bit. Maybe I'm gonna go all shrink and maybe put this on a bulleted list. Give it a little bit of differentiation. All right, great. So then we have the animal-like protists. Here's some examples. They are sort of animal-like because they consume others for food. They're heterotrophs that ingest other bacteria and algae. Right here we actually have a picture of a amoeba here. This is an amoeba that's the amoeba is circled in red here, and it's consuming a paramecium. Let me put the paramecium in green. It's consuming the paramecium. Um, this They have ciliates and amoebas and zooflagellates. These are things that have flagella on them or have cilia on them. And so here we have that example of the protozoan, and they're sort of animal-like hunters. Fungus-like protists are more fungus-like because they tend to feed on decaying matter. So they're looking for things that are already dead or things that are dying. And they absorb the nutrients straight through their cell wall as opposed to consuming and digesting. They are going to absorb nutrients by whatever they're attached to. So that's what that makes them a little bit more fungus-like. they will consume other organisms and other parasites. This includes slime molds and downy mildews. You can see a mildew here on the outside of the organism that's growing, so outside the fish. Uh, so they kind of look like funguses, so that's why. Now here's Kingdom Chromista. This is a slightly different kingdom. Was originally part of the protists, as I mentioned before. They are like the plant-like protists. They contain some sort of form of chlorophyll, which allows for photosynthesis. And this includes lots of different types of algae and water slimes, brown algae, phytoplankton. We can see the word slime here, which means that we do have sort of a photosynthetic slime mold, probably, or something similar to a photosynthetic slime mold. The presence of chlorophyll suggests that it should be distinctive. They are distinctive enough that some scientists feel they should be in their own kingdom, as we're discussing right now. Many are commonly referred to as algae, but that includes also the giant kelp. And these are multicellular. They do appear to look like plants a lot of the time, but they are actually simpler than plants. They don't usually have a lot of structure to them. They're very simple things. And so there are a couple that consume organisms or are parasites when light is unavailable, so they can actually change forms and function. Here's an example of a chromista here, an algae, where we have some form of chlorophyll use. Not a chloroplast, they just are able to produce chlorophyll, is I believe the concept here. So if we look at the protozoans and the chromistas from an evolutionary idea, there are theories that suggest they formed when large prokaryotes engulfed smaller prokaryotes. This would allow the organisms to live symbiotically and eventually evolve into a single organism where the engulfed organism was actually able to reproduce, probably at a slower rate, while the outer organism reproduced, and then its offspring would have this engulfed organism inside of it. So think about, this is a little bit hard to imagine, but you've got this bigger prokaryote, just like say that amoeba, and then it absorbs that smaller one, and then over time, this one on the inside, I'm trying to duplicates, and it undergoes asynthetic, and then this one, then the outside one duplicates, and then but there was two of these, so ends up in each. And then over time, so as long as the reproduction rates were different, you could imagine that every time that the larger cell divided, as long as the inner cell had had a chance to divide, you would be able to sort of continue to keep a one of these purple guys inside. And so that's kind of the concept, that we would have sort of different rates of cell division to produce this new species that now has, cell division now has this little organelle living inside of it, but it's not an organelle. It's another organism. Protists might have been the first eukaryotes in this way. They have now a structure inside them. Remember that prokaryotes don't have a structure, so this is the first eukaryotes. So I do have a little write-up on endosymbiosis theory. I feel like you should maybe just give this a read. But this theory has been well tested and well looked at. It certainly maps well onto the observations they've made about the rate of differences in different organisms by the number of differences in their mRNA and other things like that. But the idea is that the mitochondria and the chloroplasts were examples of these primitive bacteria-like organisms. So not just in the formation of all the organelles, but even the idea that the mitochondria and the chloroplast are our origins are this way. And the symbiosis would occur when the two different species benefited from living and working together. When one lives inside of the other, it's called endosymbiosis. And so example would be that a eukaryotic-like cell, maybe a prokaryotic organism that was just on the edge of becoming a eukaryotic one, or maybe a more eukaryotic, maybe a eukaryotic cell that didn't have a mitochondria. It got its energy in a different way, and it began as early as, this could have been as early as 2.7 billion years ago. Well, we do have examples of prokaryotic cells that are 3.8 billion years ago. So then the symbiosis theory would suggest that these complicated mitochondria and chloroplast organelles actually started off as these separate bacteria, as I suggested earlier in the previous page. The larger eukaryotic cell may have ingested this mitochondria, forming a mutually beneficial relationship. The chloroplast and mitochondria would be protected while they fed on the energy molecules. One of the things that happens in evolution is that you can become more simple if your life doesn't require complications. Like a mitochondria and a chloroplast could stop spending energy on robust cell walls and instead focus on what they're good at, which is converting energy. And the eukaryotic cell around it can stop focusing on chasing down energy continuously because it now has its own energy factory. And instead, that cell can now focus on protection and other strategies. Over millions of years, or billions of years, we should say. So let's put a B in there. Billions of years, the mitochondria, the chloroplast, became the specialized tools we see today. And now you cannot live outside the cell, although that's not true. Not true. They have now, just recently, like within the last year, they have now observed, scientists, I should really type these things, but scientists have now uh, observed mitochondria free floating. Not true. Free floating mitochondria have recently been detected in blood of animals. So those mitochondria can actually survive outside and freely moving around, and this could actually be another way that that genetic differences have developed. Anyways, there's some evidence for this. There's mitochondria and chloroplasts that have striking similarities to bacteria cells. They also have their own DNA that's separate, or their own mitochondrial information. Both organelles use their DNA to produce proteins and enzymes they require. They have a double membrane, just like a regular cell does. And the two organelles also reproduce like bacteria, replicating their own DNA and then directing their own division. It's not something the cell does. Mitochondrial DNA also has a unique pattern of inheritance. It is passed down directly from mother to child, so it accumulates changes much more slowly than other DNA. Because of its unique characteristics, mitochondrial DNA has provided important clues about evolutionary history, and these differences are used to estimate how closely related species are in typical evolutionary biology. So yeah, I think I'm going to talk more about where the mitochondria come from at a later date, but they are directly from your mother. So all of your mitochondria and a lot of your actual proteins and structures and things in your cells is from your mother because your mother donated the egg that led to your birth. So your biological parent there, your biological mother, will be the egg source, or your biological egg source will be the unique provider of your mitochondria. They were in the egg first. The sperm actually has none of that. The sperm is just a genetic DNA donor. It has a few proteins and some enzymes and some things that it brings with it, but typically we see the sperm as just the genetic information donor. Meanwhile, the mitochondria and some of those important proteins and enzymes needed to start life are only from the mother. So here's an example of what this might look like. You have a young Earth prokaryote. We have one that breathes oxygen, one that breathes bacteria. You get a membrane enclosed organelles that start to form as one of these prokaryotes, the larger one, starts to develop folds in its membrane. While these folds are developing, it also snatches up one of these smaller oxygen-breathing bacterium. Those bacterium become smaller and more diverse and more distinct from their free-floating counterparts as the cell develops. Over time, the cell develops develops various features, including the vacuole for the plant cell that will help feed that chloroplast, that photosynthetic bacterium. And the modern animal cell also develops a much smaller, but allows for many more of these mitochondria to form as well as the other organelles form. All right, this video has gone on long enough. I think we're going to come back to cover the fungi, the plants, and the animals in our next video. Thanks for your attention and have a great day.